All right, so it's a pleasure to be here. I'm sorry I'm a little bit late because I'm also a clinical geneticist on top of everything else, so I was seeing patients this morning. So I'm talking a little bit, and so I kind of missed what you heard about this morning, but certainly this talk is going to be about kind of implementing whole exome sequencing kind of in the real world, which is what we're trying to do with this project that we call the BabySeq project. So the BabySeq project is, the actual name of it is Genomic Sequencing for Childhood Risk and Newborn Illness, and it's part of a consortium called NSIGHT. And what happened is about 5 years ago, the Child Institute at the NIH, so NICHD, and the Genomics Institute, the NHGRI, kind of got together to and put out an RFA for groups to study how we would potentially integrate genomic sequencing into newborn screening. And it really came from this idea that, kind of Francis Collins in particular has talked about is, at some point, everybody in the next few years, everybody's going to have their genome sequenced or their exome sequenced. And so this is looking at that really in the newborn period. So there's four sites, and so we're the site here, the Brigham, Boston Children's, and Baylor Medical College. And each site is doing something quite different, and I'm going to talk about what our site is. So we call it the BabySeq project. It's kind of a cute name. And the idea is really to examine the clinical utility and impact of genomic sequencing in newborns. And so we're looking at kind of the medical impact. So what is the impact on in the individual and on public health? Looking at behavioral impact, what is the impact on physicians and patient behavior from kind of getting this information in the newborn period. And then looking at the economic impact, so what is the kind of the cost in terms of the healthcare system. So this is very ambitious. I'm not going to say that we're really able to look at all these, but at least I think we've started to try to kind of answer some of these questions. So this is what the BabySeq project looks like. So it's kind of our consort diagram. So we enroll infants in the intensive care unit at the Brigham and Women's Hospital, the Boston Children's Hospital, and the MGH. And then we have a cohort of well newborns from the Brigham, which is a large well baby nursery. And we randomize within each cohort to either standard of care, newborn screening and family history, in one arm, and the other arm is the same thing, but then with a genome report, and if there's indication, what we call indication-based analysis, which I'll get to. And then essentially we then do a disclosure where we sit down with the families and in both arms, and we talk about kind of what we found in the genome arm and the arm that didn't get sequencing. We really kind of go through their family history and what kind of things that they're at risk for. And then we put the information into the medical records, and we send the report to their pediatricians. And then we do a series of kind of patient and physician surveys to look at this question of kind of what the impact is on families. So we have two reporting strategies. So everybody who's in the sequencing arm gets this newborn genomic sequencing report, or NGSR, which has risk for childhood onset conditions and a few highly actionable adult onset only conditions. Carrier status for childhood disease and pharmacogenomics relevant to pediatrics. And then in children, particularly in the NICU, who have a particular problem, so for example, a child who has seizures, they would get an indication-based report with genes associated with their clinical features. But since we're already kind of giving that back in this report, the question is what's different here, and essentially we kind of do a little bit of a deeper dive. We might look into some genes that we didn't look at in the regular report, and we report, in these cases where there's a higher probability, we may report VUSs, whereas in the others we really just report pathogenic and likely pathogenic variants. So these are kind of criteria. So again, childhood onset, highly penetrant disorders, childhood actionable and at least moderately penetrant, and then we have a few adult onset only highly penetrant, highly actionable conditions, and right now that really includes B or C, the BRCA genes, as well as Lynch syndrome. So these are kind of from the ACMG's list of returnable genes that are just adult onset. And then again, in the general report, just pathogenic or likely pathogenic variants, and if there's a high prior probability in the indication-based report, we go down to potentially reporting VUSs. So this is just to give you a sense of what we're reporting compared to what happens in newborn screening now. Now, I know most of you probably don't know a lot about newborn screening, but essentially there's something called the RUSP, which is kind of the conditions that really pretty much all states include, and then some states include others and more. But on the kind of conditions that are on that kind of across the board newborn screening, with the heel prick at birth, there's 34 conditions that are reported. The American College of Medical Genetics has their now ACMG 59 list of secondary findings that should be reported or offered to be reported to anybody who has genome sequencing for any other primary indication, and that includes 27 conditions. In BabySeq, we're talking about like over 1,000 conditions. So this is not just highly actionable things. It's any kind of adult childhood onset condition, as well as things that are actionable. And actionability, or in other words, ability to do something about it, is not actually a criteria for us reporting. So this would, if something like this was incorporated into newborn screening, it would make a big difference, because it's really a lot of things now that are not currently on newborn screening. So this just shows you, it's probably a little bit hard to see because of the light, but essentially the idea here is that we're doing surveys. And that's how we're looking at really, so kind of Jeff talked about this, ELSI, so we talk about ethical, legal, social implications. It's called ELSI, it's actually a branch of the NHGRI. When we talk about ELSI, we talk about kind of what happens to people, how they, what the impact on individuals are of getting this type of information. And that's what these surveys are designed to do. So the parents all do a survey, a baseline survey, so we're trying to get what their views are towards a lot of things, including genetics and newborn screen at baseline. We do a survey right after we do our disclosure. So again, we sit down with all the families, do this disclosure, whether they're in the control or the sequencing arm. And then they get a survey immediately afterwards. We do another survey at 3 months and another survey at 10 months. In physicians who are receiving these, we give them do a baseline survey, and then a survey after they get the results back. So once we've sat down with the families, we write up a letter, we send the results to the pediatricians, and they get a survey. And then at the end of the study, which is coming up in a few months, actually, we'll be doing surveys at the end of the study of physicians. So I'm going to talk a little bit about enrollment. And the reason I'm talking about it is that I think what I was kind of asked to do is talk a little bit about challenges. And as it turns out, enrollment was a big challenge for us. So when we, prior to doing this study, we did a smaller study where we went to families in the Brigham Well Baby Nursery and said, if you had an option to get your baby in a study of whole exome sequencing at birth, would you enroll? And it was something like 75% of them said that they would, either most likely or definitely would enroll in such a study. So we thought, this is great. We're going to really have a lot of people kind of interested in enrolling in our study. And it didn't actually turn out that way. So one of the lessons, if you're doing any kind of enrolling patients, there's a difference between what patients say they're going to do and what patients actually do when they're faced with a situation. So in the healthy cohort, we literally have approached 4,000 families over the past 2 or 3 years. And we first approach them and we just go in there and say, would you be interested in hearing more about a study of sequencing in the newborn? Do you want to hear about the study? And 9% of them said yes. So that's the first thing. So I think part of what's going on here is that, first of all, people are not necessarily like upfront. They even want to hear about our study. And there's a lot of other reasons. They just had a baby. I don't know if any of you had a baby, but it's kind of a busy time. But it's a busy time. They're kind of, there's a lot going on. So 9% of them did not even want to hear about our study in the first place. However, when we got to the point, so what we did is we asked if they're interested, and then if they're interested, we set up this consent session where we sit down with them, we go through the consent form and really explain the study. So of these, 4,000, about 400 of them went to the consent session. And then, once they heard about a study, really a lot of them enrolled. So about 64% of them enrolled in the study. So it was really, once they heard about it, they were interested, but before they heard about it, they really weren't. And there was a similar thing in the intensive care unit. And we were actually really surprised about the ICUs, because the ICUs are full of kids who have things that have some kind of maybe genetic. Now, granted, there are a lot of kids who are, premature births and this kind of thing, they're in intensive care unit. But we thought we'd have a really big uptake because people would think, wow, this is great, I'm going to get sequencing, potentially get sequencing for free, realizing they're being randomized. But it was the same thing. It was a little bit higher, so 16% went on to the consent session. But again, once they got to the consent session, they really heard about all the risks and benefits and all sorts of, we have to put in all sorts of potentially bad things that could happen. Really, a lot of them enrolled. So that was kind of the first lesson we learned from this, is that what people say they're going to do, what they do ends up being a little bit different. So then about halfway through the study, we realized a lot of people weren't enrolling. So we tried to understand why people weren't enrolling. So this is kind of the whole, cohort together. Again, it's not everybody because we started about halfway through. But it really, the majority of people did not enroll because they were just not interested in research. And that was most of the people kind of in the beginning. And then some people we didn't get a reason for, but then other reasons are kind of laid out here. So study logistics was a big reason, worry about return of results, not comfortable with sequencing, privacy discrimination issues, and just feeling overwhelmed. And so these were kind of some of the reasons why people said they didn't want to enroll. So then we looked at and kind of compared the decliners, the people who the decliners pre means the people that we just said, do you want to hear about the study? They said no, to the people who declined after hearing about the consent. And what you see is that the pre people was really that they were not interested in sequencing, they were overwhelmed, and they were concerned about the study design and logistics. And those were really the main reasons. And a lot of it was just they didn't want to be randomized, they didn't want to have to do surveys, they just didn't want to kind of have to deal with it. Whereas once we got to really talking about the study, it was more about issues about privacy and discrimination and return of results. And so I think it's kind of interesting to see why people do and do not enroll, and what kind of, we talk about sequencing as if why wouldn't everybody want to get sequenced, but look at this, there really are issues that people have when they think about reasons for not participating in this type of study. Okay, so in summary, the major initial barriers are lack of interest in research, logistic concerns, likely, and I think this is also true. The beginning, why they didn't want to hear about it, it wasn't even so much our study, they didn't really want to hear about any study. So it's part of it is trying to enroll people at this time where they've just had a baby, they're in the hospital for 2 days, they're out the door, and they're kind of overwhelmed. So the impact of the consent education sessions kind of brought out concerns about privacy, result, and implications. And understanding, I think, parental concerns around newborn genomic research may inform fetal utilization. And I think it just gets the issue about that we really need to, if we want to do research or even do any kind of other things in newborn screening, we really kind of understand what kind of concerns families have and really start to deal with those if we're going to move forward in this type of research. Okay, so then I'm going to talk a little bit about our monogenic findings. And I'm first going to say, before I start, is just to kind of to set up for you. So we're doing screening sequencing. There's no prior probability, for most of these, there's no prior probability that there's a problem. And so what we realized as we started in this project is that if we're only enrolling, going to be sequencing, 150 to 200 kids, there's maybe not a lot of things we're going to find. And so at that point, we actually added carrier status. And so our study now includes both monogenic findings and carrier status because we want to return something to people and see how they would respond to that. And we felt that carrier status would also allow us to look at how people respond to carrier status. So there's kind of two parts of our returning findings, and the first part is these monogenic findings, which again was the kind of primary role of the study. So we ended up about 160 infants were sequenced, and out of those infants, again, over 1,000 conditions and genes we're looking at, we had 18 cases, or 11%, that had some finding predictive of a monogenic disease, and then 89% with no findings or only carrier status variants. So if you break down those 18 cases, there were 4 that had diagnostic findings and then 14 that had findings that were potentially kind of predictive, and I'm going to go into a little bit of detail about one of those. But of all of the people that we sequenced, only 1 had a finding that explained a known kind of condition that they had. And that condition was in a baby, and again, I'm trying not to be too kind of medical, but basically an infant that has something that disease that's called VACTERL. So it's a combination of features. And so the baby had an imperfect anterior displaced anus, hydronephrosis, reflux, kidney issues, mildly dysmorphic, strabismus, and some other things. So these were the features that the child had. And we found this frameshift truncated mutation in ANKRD11. And this is associated with something called KBG syndrome. And I kept saying KGB, but it's not like the Russian KGB, it's KBG. But anyway, this is a syndrome that has features that really were not consistent with what the child had. So this gets, again, into this kind of prior probability. The child has something that looks kind of genetic. We found something that didn't really quite fit. And one of the things that happened is that then we originally thought, well, maybe this doesn't really explain it. But as it turns out, when we looked further, some of the features that this baby had were actually found in this syndrome. And so maybe that we've now kind of expanded the phenotype, which is called phenotype expansion, of this syndrome based on these findings. And one of the things that happened with the family is that they're now, for them, they kind of have an explanation for why their child had all of these problems. And so now they're part of a family group of other families with KBG syndrome. So this was kind of an interesting, because it got at the issue of you find something that doesn't quite fit, and maybe we're actually expanding the phenotype for some of these disorders. Okay, so then findings prompting the discovery of an unrecognized phenotype. So this was kind of interesting. This is a healthy baby that had a paternally inherited nonsense mutation in a gene called ELN. And ELN, it stands for elfin, and it's because it's associated with something called Williams syndrome, where one of the features is that children have kind of elfin-like faces. That's how that name came about. But it's associated, a part of Williams syndrome, and this gene in particular, is supravalvular aortic stenosis. And so this was a healthy baby. We found this, and so we did echocardiograms in the father who had this and in the baby. And as it turns out, the baby actually did have kind of mild hypoplasia in that supravalvular aortic region. So it turns out we didn't know anything about it. We did this finding, we looked at the baby, and we actually saw something. The father, on the other hand, who also had this, was normal. So it's clearly not a completely penetrant disorder. And this was also very interesting. So this was a healthy baby that was a compound heterozygous for mutations in BTD. So this is a gene that's associated with something called biotinidase deficiency. So biotinidase deficiency is an enzyme deficiency. It's actually on the newborn screen. And what happened is that the baby's initial newborn screen was positive for biotinidase deficiency, but kind of barely, and then they repeated it, and it was normal. So after the newborn screen, they said the baby doesn't have this biotinidase deficiency. And then we found this, and we on our screen, and so we sent it to a metabolism clinic, and as it turns out, when you actually measure, not just on a screening, but you actually go in the clinic and measure biotinidase levels, they actually are low. And so the baby probably has this mild biotinidase deficiency. And the nice thing about it is you can do something about it. You can give, biotin. I think it's biotin is what they give. Yeah, biotin. So you can actually treat it. So now the baby's on biotin. Now we don't know if the baby would have gone and have a normal, happy life without ever getting biotin and would have been totally fine or not. But we did find this, and we are able to do something about it. This is, so now kind of looking at findings predicting future disease risk. So this was a healthy baby that had maternally inherited NYBPC3 mutation, which is associated with hypertrophic cardiomyopathy. And so it was no reported cardiomyopathy in the family, and so we're doing kind of a subsequent cardiac workup. So far, the mother doesn't have it, and the baby hasn't been evaluated. So we'll have to see if this really kind of pans out to be really totally pathogenic. So then there are some future disease risk where there was a positive family history. So as I told you, we had added, so we did have a few kind of adult onset only conditions, and one of them is BRCA. And it turns out there was two individuals with variants in BRCA2, and then one in the MSH2, the Lynch syndrome gene. And as it turns out that the BRCA mutations were distant relatives with breast and ovarian cancer. So this ended up being very interesting because there was no kind of immediate history of breast cancer or ovarian cancer in any of the families. But when you looked back further through the paternal lines, there were other kind of aunts of the mother, because in these cases the mother was a carrier. And so this really did make a difference to the mother because now the moms were being tested, and in at least one of the cases, the child who had it was a boy who subsequently passed away, but the others did have, it was really informative for the family. And then the MSH2 variant, that was a maternal uncle, this was through the mother who had gastric cancer, and so it's been kind of, follow-up has been kind of satisfying with that. And I'm going to skip through these since I don't have as much time, but there's some other kind of interesting things that we've found where there was no family history. So just briefly to talk about carrier status, and then I'll probably end because I think I'm coming to the end of my time. So carrier status is a different story because most of us are carriers. We always say, 3 to 4 kind of rare recessive syndromes. We found on average carrier status for 2 conditions per infant. And this just shows kind of the screen. So most of them either had 1 or 2, and then some had more. And these are what some of the findings are. But I just want to talk about a case example because I think this is kind of shows how this type of screening can be useful. So we found this one family here. We found 4 carrier status for 4 different variants. So I'm not going to list it there, but there's 4 different variants. And so the family, because there was 4 of them, and the thing is that the baby has it and got it from one, but you never know what the other parent has, and you kind of assume they don't, probably don't have anything, but you never know because these are all rare. So it turns out that both parents were carriers for one of these variants, which is this is a nephropathic cystinosis variant. And so what happened then from this is that we identified the carrier status in the child. We don't check the parents for carrier status. It's not part of the study. They went and got prenatal testing. As it turns out, they both were carriers for this. And so this shows how there was some kind of utility from the perspective of the family regarding the carrier status. This is just kind of summarizing the outcomes, but of the sequencing part of it. So again, about 11% of babies had genomic findings that were diagnostic or predictive, and this often prompted family members who may have the same condition in those families. And I think I'm going to stop there because I'm probably kind of out of time, but I'm happy to take any questions.
You did finish a couple minutes early. So
okay, good.
Yeah, so any questions for Ingrid? Fascinating project. Good.
Yeah, wonderful project, very interesting. I think it clearly shows how there's applications, if this were expanded, both to complement the existing newborn screening programs, and maybe catch things that they wouldn't catch, or help explain more quickly the things they do catch, and also to be able to catch things that aren't easily or included as part of the newborn screening. So how do you see this expanding? What are the challenges to expanding this so that, for example, some sort of exome or genome sequencing would be done alongside of newborns, the traditional newborn screening?
Right. So first, I want to say that you're absolutely right about this couldn't replace it because there's some things like congenital hypothyroidism that are not really genetic. So this, and as it turns out, there is a group that's, one of the groups that's looking at people who have a metabolic disorder, would you have picked it up on sequencing? It turns out that the metabolic mass spec is more sensitive and predictive.
There might be variants that aren't known.
Exactly, right, so it would complement. So there's a couple issues. So right now, there's a bunch of reasons why it wouldn't be incorporated. Cost is a huge one. And we really don't understand the relationship between the genes and the disorders. I mean, we don't understand penetrance. We don't understand so much, and we're seeing this here. So we have these, hypertrophic cardiomyopathy variants, and it's inherited in the family, and nobody has hypertrophic cardiomyopathy. That's because the variants were discovered in a person who had hypertrophic cardiomyopathy, and then you did the genetic test and you found this. So I think that right now, the lack of being able to make correlations between the genetics and the disorder, our lack of understanding of penetrance, and then plus the cost are kind of reasons why right now it wouldn't be good. It's a pretty good test for BRCA, but it's not a good test for the childhood conditions.
It seems like it's a little bit of a chicken and egg thing, but also I think there's conditions like SMA, for example, where I don't know that there's good biochemical screens for it,
Right.
But if you could detect it,
Oh, retinoblastoma. Yeah, there's like retinoblastoma is a great. There's some really great cancers. So there's probably a few that you could add on now that are cancer predisposition genes that are like on the ACMG 59 list, childhood onset, that would make a difference because you would screen earlier. But the rest of the genome or exome, I just don't think we know enough about it. So that's now. Fast forward 10 years, it may be different.
Question there.
Did you look at all at the diversity of population, either in specifically said about hypertrophy, whatever.
That's her presentation.
But also just in your enrollment conditions, were there certain groups that were more likely to enroll?
Oh, absolutely. Highly educated. We have geneticists who enrolled. I mean, it's just, that's a big problem with these kind of things because, so the Brigham is a very broad, well baby nursery. They have everybody there. But we really found that most people that enrolled are, white, highly educated, or individuals. It's kind of the early adopters are the ones who enroll. We do have a few Hispanic and African American enrollees, but it's by far, we didn't get that in this. And so, what we'd love to do if we could get another grant to kind of move forward is to go to, what we've talked about doing is going to clinics in diverse areas, in health centers, enrolling in the first year of life as opposed to the newborn, and try to get more of that population. But I think it's going to be harder to do. So we don't really have a well-represented population in terms of diversity.
So is there a regulation set in place to prevent insurance company buys or select against their screening results?
Well, there's laws against that. So there's a national law called GINA, the Genetics Non-Discrimination Act. So in terms of health insurance, it's not an issue because there's a law in place right now. It tends to be, and even like in Massachusetts, there's laws against discrimination in terms of disability insurance and life insurance, which is not true everywhere. But I don't know if things are going to change, but right now, you can't discriminate for health insurance based on genetics.
Anybody else? Oh, we got one more up there.
You're going to have to yell.
Okay, how successful are your treatments after you find out?
Yeah, actionable.
Yeah, so for us, the only thing that was actionable and treatable was a biotinidase deficiency. The others were looking for structural changes, so looking for cardiomyopathy, looking for supravalvular aortic stenosis. And most of them were not imminent. They're not treatable immediately. Like the newborn screen is stuff that's immediately treatable. A lot of things were more surveillance to prevent things. So that's the same thing with BRCA. It's just not actionable in childhood, where you get to be in your 20s or whatever, then you start doing screening. So it's a lot of the things that were on this were kind of screening things as opposed to treatable, the findings that we had.
What was the cost?
Oh gosh, I don't remember. I mean, it's ever the cost of sequencing is at the Broad. Essentially this was done through the LMM. And so Heidi Brim was one of the co-PIs. So, so it was clear.
A couple of years ago, so you think about where even the costs have come since
Well,
then.
we're still kind of doing it, so the costs are kind of doing it. But I mean, we're still talking about, hundreds of dollars per person, which is why, if you're going to do something in newborn screening and you're going to screen millions of babies, you need something that's really cost effective. And then it's not just the cost of it. So we do the sequencing. It's like in a CLIA lab, it's sequenced. But then our team does the analysis of it. And that doesn't cost us anything. It's part of our team. But that is also part of the cost of the whole sequencing.